Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to talk about how sodium chloride is reabsorbed in the proximal tubule. So with that, let's give it a go. So the first thing that we're going to talk about is the attributes of the proximal tubule, so the characteristics of it. And the reason why is because in order to understand how sodium chloride is reabsorbed, you have to know the certain characteristics and attributes of the tubule. So the first attribute is that the proximal tubule has extensive membrane infoldings. And these membrane infoldings are present on the apical membrane, and they're referred to as the brush border. So the brush border serves to basically greatly increase the surface area of the cell membrane. The second attribute is that the proximal tubule has a lot of mitochondria in it. This is going to allow the cell to consume a lot of oxygen and therefore produce a lot of ATP. This ATP is going to drive the action of the sodium-potassium pump, which we're going to talk about, and this is going to allow sodium chloride to be absorbed at a high rate, and it's going to power a lot of the mechanisms that we're going to talk about today. The third attribute is that the proximal tubule is leaky, and because of its leakiness, it has a very low transepithelial voltage, it has a low resistance, and in addition, it's going to allow certain paracellular mechanisms to take place for the solutes. And then the last attribute is that the proximal tubule is characterized by non-selective tight junctions. So now that we know these attributes, let's talk about how sodium chloride is reabsorbed in the proximal tubule. So in order to understand how sodium chloride is reabsorbed by the proximal tubule, let's draw the diagram that we're going to use today. So the first thing that we're going to draw is we're going to draw our cells. And these cells, as we talked about, are epithelial cells. And this is going to be the proximal tubule region of the nephron. So we basically have two sides on the screen. The first side is going to be the tubule lumen. So this is going to be inside the nephron. And then we're also going to have the interstitial space, which is outside the nephron. So please keep these two spaces organized because certain transporters are going to be on certain sides of the cell. So let's talk about first how sodium is absorbed in the proximal tubule. Now the proximal tubule is divided into the early and late proximal tubule, and the early proximal tubule is going to be segments 1 and 2, and the late is going to be segment 3. So this mechanism that we're going to be talking about here is going to be present in all segments of the proximal tubule, so both the early and late proximal tubule. So let's take a look at what this first mechanism is. So the first important part of this mechanism is going to be the sodium-potassium pump. So the sodium-potassium pump is basically going to pump 3 sodiums out of the cell into the interstitial space, and then 2 potassiums into the cell from the interstitial space. And it accomplishes this by hydrolyzing ATP and using that energy from the hydrolysis to drive this pumping action. So this action is going to be very important for the rest of the mechanism, because on the apical membrane facing the tubule lumen, we have a specific secondary active transporter. And this secondary active transporter is the sodium hydrogen ion exchange protein, specifically NHE3. So what this protein does is it basically couples the downward movement of sodium to the uphill movement of hydronium ions. So basically what it does is it brings sodium down its electrochemical gradient into the cell, uses that energy in order to pump hydronium ions against its gradient out into the tubule lumen. So once again, this exchange protein basically moves sodium down its electrochemical gradient, which releases energy, which allows the exchange protein to move hydronium ions against its gradient into the tubule lumen. So this is going to be the main way in which sodium is reabsorbed in this mechanism. So after the hydronium ion is brought into the tubule lumen, it is going to combine with bicarbonate in the tubule, and these two are going to form carbonic acid. So carbonic acid is then going to be converted into 2 molecules. And this conversion is going to take place via carbonic anhydrase 4, which is present on the brush border membrane. So what we see here is that this enzyme is actually connected to the cell membrane. So what this enzyme does is it basically catalyzes the conversion of carbonic acid into water and carbon dioxide. Now, in order to get the water into the cell, water is going to move through aquaporin 1. So it moves through aquaporin 1 to go into the cell. Now, carbon dioxide can get into the cell via two mechanisms. The first mechanism is through passive transport by diffusing through the cell membrane, and the second way is actually it can move through aquaporin 1. So now that we have both water and carbon dioxide in the cell, the two are going to be reacted together through the enzyme carbonic anhydrase 2. So carbonic anhydrase 2 basically catalyzes the conversion of water and carbon dioxide into carbonic acid once again. Carbonic acid then dissociates into protons, which helps feed into this exchange protein. And then the other part of it, the bicarbonate, is going to be used in another secondary active transporter. And this secondary active transporter is going to be the sodium bicarbonate co-transporter. And it's present right here, facing the interstitial space. So what this particular co-transporter does is it basically co-transports 3 bicarbonate molecules along with 1 sodium molecule. So it has a 3-to-1 ratio, so 3 bicarbonates to 1 sodium. So basically, the co-transport of these two things allows sodium to move into the interstitial space and therefore be reabsorbed. So this is going to be the first mechanism by which sodium is reabsorbed. So the water that did enter into the cell can then leave the cell through aquaporin 1 present on the basolateral membrane, therefore going into the interstitial space. So that was how sodium was reabsorbed. So let's now talk about how chloride is reabsorbed in the proximal tubule. So the first region of the proximal tubule that we're going to look at is the early proximal tubule, specifically segments 1 and 2. So remember the last few minutes of the video that sodium has been reabsorbed by the proximal tubule. It's being reabsorbed as we go down the tubule. Now, as a result of this, what happens is positive charge is leaving the tubule and entering into the interstitial space. As a result, the tubule lumen is becoming more negatively charged. And in fact, it reaches a lumen voltage of around -2 to -4 millivolts. So the voltage inside the tubule lumen is going to be negative. So in addition to this, we also had water being reabsorbed by the cells, right? So water is being reabsorbed. And as a result, we have 2 factors that are going to drive chloride reabsorption. The first factor is that we have the negative charge that is going to repel chloride. And then also we have water being reabsorbed, which increases the concentration of chloride in the tubule. So these 2 mechanisms work together in order to drive the paracellular transport of chloride across the wall into the interstitial space. Now remember that chloride can be transported paracellularly because the proximal tubule is very leaky. So it's both the negative voltage and the increasing chloride concentration that drives the reabsorption through this mechanism. So this mechanism is going to take place in the early proximal tubule, specifically segments 1 and 2. So now let's talk about how chloride is going to be reabsorbed in the late proximal tubule, specifically segment 3. So in order to get this going, we need to have our sodium-potassium pump. So the sodium-potassium pump pumps 3 sodiums out of the cell into the interstitial space and 2 potassiums into the cell. And it does this by hydrolyzing ATP and utilizing that energy to accomplish this. So an important protein that is responsible for transcellular chloride reabsorption is going to be the sodium hydronium exchange protein. So remember that this protein is basically going to couple the downhill movement of sodium to the uphill movement of hydronium. So it uses the energy released from moving sodium into the cell to basically move hydronium out of the cell into the tubule lumen. Now the second important protein is going to be the anion exchange protein. So the anion exchange protein is going to exchange one anion for another. So specifically, it's going to exchange one anion by transporting it from inside the cell to outside. In this case, it's using bicarbonate, but it can use other anions, what it's going to do is it's going to couple that movement to the movement of chloride into the cell. So chloride is going to move into the cell and other anions are going to move out. So this is how chloride is going to be reabsorbed transcellularly from the tubule lumen. Now, after this occurs, the hydronium ion and the bicarbonate ion are going to combine together to form carbonic acid. Carbonic acid is then going to break down into water and carbon dioxide through carbonic anhydrase, which is present on the cell membrane. Then water and carbon dioxide will move into the cell through the aquaporin 1 protein. Now, after they enter into the cell, these two molecules are going to react via the other carbonic anhydrase protein in the cell to form carbonic acid. Carbonic acid then dissociates, feeding both of these exchange proteins here. So once we have chlorine in the cell, how are we going to get the chloride into the interstitial space? Well, the way that we're going to do it is we're going to use the potassium chloride cotransporter. So basically what it does is it basically couples the downhill movement of potassium to the uphill movement of chloride. So remember that the potassium wants to move out of the cell. So when this cotransporter moves potassium out of the cell, this releases energy, allowing the transporter to transport chloride out of the cell into the interstitial space. So this is going to be the transcellular mechanism in how chloride is going to move into the interstitial space. So in addition to these transcellular mechanisms in the late proximal tubule, we also have some paracellular mechanisms. So what we see here is that chloride is being reabsorbed transcellularly. As a result, this causes the voltage of the late proximal tubule to actually increase. So it increases to around +2 to +4 millivolts. So as a result, the voltage in the tubule is positive. This is actually going to drive the paracellular movement of sodium from the tubule into the interstitial space. However, remember that we also have water being reabsorbed in the tubule. So as a result, the chloride concentration is increasing. So even though the voltage is positive, because we have an increased chloride concentration, chloride is also going to move down its chemical gradient into the interstitial space through the paracellular lute. So in the late proximal tubule, we have both a transcellular and a paracellular mechanism working in order to reabsorb chloride. So the last mechanism in how sodium and chloride are reabsorbed is something called solvent drag. So because of the reabsorption of both sodium bicarbonate and sodium chloride, this is going to create an osmotic gradient. And this osmotic gradient is going to favor the movement of water from the tubule lumen into the interstitial space. And this occurs by water moving through the paracellular roots, so in between the adjacent cells. Now, as water, the solvent, moves from the tubule into the interstitial space, this is actually going to drag the cations and anions with it. So it drags both chloride and sodium with it. And this is what we call solvent drag. Water, the solvent, is dragging with it both sodium and chloride, allowing sodium and chloride to be reabsorbed. So this is another mechanism in which how sodium and chloride are reabsorbed in the proximal tubule. So remember that inside the proximal tubule cells, we have a lot of mitochondria. So as a result, these cells basically consume a lot of oxygen. So what I would like to do is to show you the relationship between oxygen consumption and the reabsorption of sodium. So the first thing to take note of is that the proximal tubule epithelial cells have a basal oxygen consumption. So this is the amount of oxygen they consume when they're not transporting sodium. So this is going to be the basal oxygen consumption. So what happens to the oxygen consumption as we reabsorb sodium? Well, what we see is a linear relationship. As we reabsorb more and more sodium, the oxygen consumption climbs. And the reason why is because we have a lot of mitochondria in these epithelial cells. These mitochondria consume oxygen to produce ATP, and this ATP then fuels the sodium-potassium pump, which is the main driver of all the transcellular mechanisms we talked about today. So that's it for this video. I hope it helped you understand how both sodium and chloride are reabsorbed by the proximal tubule, and I hope to see you in the next one. I apologize for this video being longer, but it is a video with a lot of different concepts. And if you're going to study for this, if you're having a quiz or exam that is going to test you on the different transporters, my recommendation is to practice drawing these mechanisms on a whiteboard or on a sheet of paper over and over and over again so that you can remember how these things work, because it can get confusing remembering where the transporters are on the epithelial cells and which segments have which transporters and so forth. So that's my study tip for you today. So once again, I thank you for watching, and I hope to see you next time.